for these companies. But I want to say that although I'm doing some research with Novartis, I have received and I will not receive any personal funds because I'm going to be talking about a Novartis drug, LCZ696, and I want to make it clear that I have no financial connection to them. So my objective this evening is I have two goals: to present the history of the physiological and clinical advances that led to the development of this first ARNi. ARNi stands for angiotensin receptor blocker neprilysin inhibitor. It's much easier to say ARNi than to say these 6 words. And secondly, to use this achievement, which I think is important in heart failure and actually for cardiology as a whole, as a case study that demonstrates the real necessity of efforts of both academia and industry to advance science. So the story really began on March 31, 2014, so this would be 27 months ago, when the Data Safety Monitoring Board stopped a clinical trial called Paradigm Heart Failure because of overwhelming efficacy in heart failure. We haven't seen that happen for a long, long time. We've seen a lot of trials stop because of adverse effects. We've seen a lot of trials, heart failure stop because of futility. But I don't know if there have been any others that have been stopped because of overwhelming efficacy. And then in September at the European Society of Cardiology, this paper was presented, and it was published simultaneously in the New England Journal of Medicine. And this angiotensin receptor neprilysin inhibitor, ARNi, angiotensin neprilysin inhibition against enalapril in heart failure. And the lead author was John McMurray from Glasgow. And we're proud at the Brigham that 2 of the co-authors, Dr. Desai and Dr. Solomon, and I know Scott Solomon will be speaking to you. I hope he won't be saying the same things that I'm saying, but made important contributions to this paper. So this is the key figure. And now I have a symbol at the top right, and A means academic, and I means industry. And this is an AI. So this clinical trial is a combined effort of an academic group, because the authors of this paper, 16 authors, 15 were academic, and 1 was an industry representative, a very, very strong person, Dr. Lefkowitz. But this was an effort between the 2 because the drug was developed by industry, and I'll show you how it was developed. So the goal of the trial was to see whether they could improve on a drug that we all have used, or a class of drugs that we have all used in the treatment of heart failure for 25 years, an ACE inhibitor, enalapril. And as you can see, the primary endpoint is cardiovascular death or heart failure hospitalization. And you can see that there's a 20% reduction. There is a hazard ratio of 0.8. And there's an obscene number of zeros after the p-value. So it's extremely positive. Now, one of the interesting things about this trial is they didn't show changes because they got this result in part, not only because the drug is good, but they had 88,300 patients. So this is the largest heart failure trial in history. So if they had 2,000 patients in this trial, it would have been borderline. So that's an important lesson. So here are the things that these benefits were consistent. And I repeat to you, I have not received any money from Novartis to say this. So compared with the enalapril, patients on LCZ were less likely to die of a cardiovascular cause or to die for any reason. Total mortality was reduced. To die suddenly, to be hospitalized for heart failure or for any reason, any cause of hospitalization, to have a visit to an emergency room, to be admitted to an ICU, to need inotropic therapy, to require devices like a left ventricular assist device, surgery for end-stage heart failure, to show deterioration in renal function, and to show biomarker evidence of myocyte injury that is release of low concentrations of component. So that's a pretty good score. Now, you always wonder when people have an interest in something, even though it's a blinded trial, what will other people think about it? Well, we know the editors of the New England Journal thought it was very good, but there are three groups that affirm the superiority who had nothing to do with the trial. The first one I've already mentioned, March 31, an independent data safety monitoring board recommended stopping the trial for overwhelming superiority. These are people who had nothing to do except to protect the patient. On August 12th last year, the FDA approved this drug for the treatment of heart failure and reduced ejection fraction. And it was a very speedy approval. Usually the FDA takes about 15 months to approve a drug, and they call in committees, and they did not call in an outside committee. They made the decision themselves, and it took 5 months to get this approval. And then thirdly, most recently, just a month ago, on May 20th, the ACC, the American Heart Association, and the Heart Failure Society of America published a focused update for heart failure, and they gave it a Class 1 recommendation. Now it's a 1B, not a 1A, because to get a 1A you have to have multiple trials, and a 1B is a single trial, and R means randomized. And as many of you know, the European Heart Failure Foundation also gave a similar reading on the same date, on May 20th. So these are really separate agreements that this is for real. So the question is, how did we get here? And what was the path? Well, it began with a professor of physiology by the name of Robert Tigerstedt, working at the Karolinska Institute in Stockholm. Now, I read his biography, and it was interesting. He was not Swedish. He came from Finland, and why would a Finnish physiologist work at the Karolinska? It's because he had a fight with his chief, and he had to go to an outlying country. And so Tigerstedt conducted an extraordinary experiment. You can see his first paper was published in 1898, and he describes it. And the key paper, the key experiment is shown as the lowest curve. So this is rabbit's blood pressure. So he took a rabbit kidney and he pulverized it in water, and he injected it into the jugular vein of another rabbit. And as you can see, as written on top, within 80 seconds, there was a rise in the mean arterial pressure, and increased by about 50%. And the other three curves, he repeated the experiment and plotted the results on the same graphic. So this was a consistent experiment. And he worked with Bergman, who was never heard from again. He was a medical student who went into primary care practice, but he got his name on one of the most important papers at the turn of the century. And Tigerstedt named this substance renin. And of course we know that renin is an enzyme, it's a protease, but of course Tigerstedt didn't know that. And I give that an A, because Tigerstedt was not working in the big industry, and he was an academic leader. And to show you how slowly things moved, nothing much happened in this, because actually this is a pretty remarkable finding, and you would think people would jump on it, but they didn't. And nothing happens until this man, Harry Goldblatt, who was a professor of pathology at Case Western Reserve University in Cleveland, and he was working with dogs. And one of his, and the experiment that he did is he produced ischemia in one renal artery, not occlusion, but ischemia. And here's the key paper. So remember, Tigerstedt and Bergman, 1898, and nothing happens for 36 years. And here Harry Goldblatt, professor of pathology, shows that when he produced constriction and produced renal ischemia, the blood pressure in the dog rose and stayed elevated. And for people who've been working in this field, they call this the Goldblatt kidney, or Goldblatt hypertension. And of course, this made it clear, if you put these two observations together, that there must be something coming out of the kidney that is causing this increase in blood pressure. And again, we got an A there. Goldblatt was an academic. Now we move to industry, and we move to the Lilly Laboratory, and in Indianapolis, Indiana, and Irvine Page and George Helmer got a crystalline pressure substance, which they called angiotonin, which resulted from the reaction between renin and renin activator. So this was a key step forward. They now got a substance which, when properly handled, would be a powerful vasoconstrictor. And now we begin to get industry interested